You can get started. Yep. I'll be up and running.
Hi, everybody. I'm Tony Cataldo. Every time I hear Anthony, I think I've done something wrong. Come from an Italian family. We're here today to talk about EIR Biopharma. And we really focus EIR Biopharma. Martin and I have created a number of companies previously that have done very, very well. We like this industry a lot. We're focusing on age-related macular degeneration. You're going to hear about the different components of that, wet versus dry, those types of things. And that asset comes from Cornell University. And then the other asset that we have that we're also talking about that, fills our portfolio is one in glaucoma, which is from Miami University, the Bascom Eye Institute, which is known as probably the #1 eye institute in the country, which means it's very high up on there. The things that Martin and I are founders of several companies. We'll get into that later. They've achieved multi-billion-dollar market caps. It doesn't happen by accident. The things that we focus on are fundamentals. I always like to start with, it's not so important what the CEO is saying about the company. It's more important of what the data is saying. So we like data because there's no ambiguity to data. It is what it is. And that particularly FDA clinical trial data. And the other is your IP. You have to spend your money there, protect your assets, make sure that, it's defensible. So you don't go for your low bid attorney, you spend the money. We spend $1,200 an hour on attorneys and you get every penny's worth for that. The third thing, and more importantly, there's really only one customer that matters in biotech companies. It's the FDA. That's your customer. If it doesn't matter how good your IP strategy is, how great your data is, if you can't make those guys happy, you're going nowhere. So the reason we've had success is because we stick to fundamentals. We don't make excuses. All right. I've heard many excuses for failure. I've just never heard one for success. So you do the work. You got to get in front of people. It's not just Zoom calls. And you have to prosecute the assets. And most importantly, you have to have the capital. We're here like everybody else to get your attention for capital. And so I'm going to turn it over to Martin. He's going to get into the actual technologies that we have, and then I'll take it back over from then. And
slideshow on the.
Yes, I do. Can you go back one slide though? We just have to make our friends happy. This is our forward-looking statements. So
does this need to have the green light on it in order for?
Yes, it does.
Okay,
there. Okay, you can hear. So sorry, just to go over the forward-looking statements. We're going to be talking about products that are in development. And it's, what we say here, of course, is all said under the auspices of the forward-looking statement. Can we go to the next slide, please?
Yes.
So as Tony was saying, yeah, we've created a number of biotech companies that have been rather successful. One of them has an FDA-approved drug. The second one is moving towards FDA approval, should be next year, I think, this year. And then the third one is still in the clinic in some ways away. So we've worked in the field of liver disease. We worked in the fields of cancer and metabolic disease. And we became interested in eye disease. There's a major unmet medical need with eye disease, particularly when you talk about macular degeneration and glaucoma. Both of these affect the adult population and children. There's also a rare disease component in the macular degeneration segment, and it's called Stargardt's disease. It's an autosomal recessive disease that affects about 40,000 kids in the United States. The disease manifests itself by starting off when the child's about 4 years old, and you have quick progressors, and they're usually blind by the time they're 10, and then you have late progressors that are blind usually by the time they're 25. There are no cures for these diseases. As Tony mentioned, we've partnered with we've partnered with the University of Miami Bascom Eye Institute in the area of glaucoma. And glaucoma, by the way, is a neurodegenerative disease. It comes on very slowly in the early stages. It's hard for your oncologist your ophthalmologist to detect it. But nevertheless, it's something that when you go get glasses and they hit you with a little puff of air, they're testing the resiliency of the eyeball. The eyeball is like a basketball. It's inflated to a certain PSI. And when they hit you with the air, it's supposed to deflate a little bit. In many instances, it doesn't. And that's a form of increased interocular pressure, which creates damage to the optic nerve. So we're all about looking at the diseases at the back of the eye. This is the retina and the optic nerve. So just for orientation, the light comes in through the lens, it goes and hits the photoreceptor cells that convert it to energy, goes through the retina and then to the optic nerve, which transports the signal to the brain. And it is your brain that actually figures out what you're looking at. The eye is kind of like a camera, it's just collecting the image. Then our other partner is Weill Cornell Medical School, part of Cornell University in New York City. And here we're concentrating on macular degeneration, both the Stargardt's disease as well as the age-related population. As we age, our vision, as you know, starts to deteriorate. Next slide, please. So there are many causes of blindness. The main causes are here. As you can see, macular degeneration is a significant number of people worldwide. 288 million worldwide suffer from macular degeneration. When you talk about glaucoma, it's about 80 million people. And glaucoma is really the what I'll call the silent guy. It starts early, it's a long process. And unfortunately for either of these disorders, there is no cure. So the game plan, the therapeutic game plan is to manage the disease process, try to slow it down, thereby preserving quality of life for the patient. Obviously, if you would normally go blind when you're 70 years old, but you can stretch it out to 80, that's a good thing. Next slide, please. So next slide. So talk a little bit about the disease targets. So what you're looking at here is the retina, and we're looking at the dry form of macular degeneration. So there are two forms. There's the wet form, the neovascular form, and this is where you get ingrowth of blood vessels and leakage of the blood vessels to the retina. And it causes disruption of the image being processed, and it kills the retinal cells. There are drugs on the market right now, anti-VEGF antibody drugs that block the growth of blood vessels. They're known as Eylea from Regeneron and Lucentis from Genentech Roche. Collectively, they're about $10 billion a year in revenue. That's roughly 20% of the AMD market. The other 80% is pretty much unserved. Recently, in February this year, Apellis received approval for a drug, an antibody drug from called Syfovre, and its job is to block a certain pathway to slow down the rate of the so-called dry form, the atropic form of the disease. So what you see here is you see the retina. We're imaging the retina right now. We're looking at the fluorescence, and you see a bunch of light-colored speckles in there. The yellow arrows are pointing to it. This is a compound called lipofuscin. It's toxic. It's a waste product that's built up from the degradation of the membrane by the photoreceptor cells, and it builds up in the retina. The retina's job, by the way, is the waste treatment plant. It's to get rid of all the waste and out of the cell. And when this lipofuscin fills up, it kills the retinal cells. And as we age, cells generally become less efficient at metabolizing and getting rid of the waste. So the dark spots there that you see in the white arrows, that is what we call geographic atrophy. This is geographically, we look at things like, spatially, part of the retina is dead. And when retina cells die, they don't regenerate. They're kind of like nerve cells. So the game plan is to slow down, get rid of the lipofuscin, the toxic waste, and slow down this rate of geographic atrophy. Next slide, please. In glaucoma is a neurodegenerative disease. It is where your optic nerve actually and the nerve cells begin to die. So the idea here is to be able to stabilize and slow down this rate of death. So what you see on the right slide, on the left side, you see a healthy eye. And what we're looking at is the optic nerve, that little tiny spot in the back of the eye. On the right of that picture where you see the glaucoma eye, you notice that it's got a big hole there. That's because the optic nerve is gone. It's died. Okay. So what you see on the right panel is an artist's drawing, and you can see how there's a deformation in the upper picture where it's damaged optic nerve. You can see a little cup formed. And this is because the optic nerve fibers have actually died and is leaving a gaping hole. And I'll show this in another slide. Next slide, please. So next slide. So I'll actually show you some real data. So in our animal models, and by the way, we are advancing to the clinic with this, but going over the animal models. What you see here, and you look on the left side in vitro, just looking at cells, the green colored fluorescent, that's lipofuscin in retinal cells. And when you treat it with IR205 in the bottom panel, you see all the green cells are gone. That's because IR205 is like a little vacuum cleaner. It goes in there and scarfs up all the lipofuscin and brings it over to something called the lysozyme in the cell, and it's processed and excreted as waste. Then if you look on the right side, this is the mouse. So, we treat the mice, and I call your attention to the lower right-hand panel. You see these two histology slides. And on the left, you see the control. You see all the orange-yellow color. That's lipofuscin built up in the cell. And you see after treatment in the mice that it's all gone. So this data demonstrates efficacy of the product sufficient to move it forward with the FDA and begin human clinical studies. Next slide. But like I say, it's not enough to just capture the lipofuscin and get it to the retina. You actually have to get the lysosome, which has died in the cell. This is a little waste treatment plant. The lipofuscin permeates the membrane and all the digestive contents leach out, and it's killing the retinal cells. So you have to restart the lysozyme. And what you see here is untreated panel on the left, and there's no red color. This red color is a dye that we use to monitor whether or not a lysosomes can excrete the waste products. Are they healthy? Do they come back alive? And you can see in the untreated, they're dead. Onomycin is a drug standard we use that is classically used to demonstrate whether or not the lysozyme is functioning. And you can see it's just a little bit of performance there with a little bit of red. But when we treat it with IR205, you can see the dramatic increase. It means all the lysozymes are being restored. The holes in the membrane have healed, and we're back in business in that the retinal cells are now functioning correctly. So this data and other data that we have is quite exciting, and moving it into the human experience, we think that it will translate to the human experience, and we'll see some improvement here, not only in the Stargardt's patients, but hopefully in the adult population too. So I'm going to transition to glaucoma. Next slide, please. So as I mentioned, glaucoma is a neurodegenerative disease. And what we have happening here is the optic nerve is being killed, is dying. The cells, the retinal ganglia cells are dying. And what you see is these green lines. These are an artist's rendition of the long axome fibers that stretch into the brain, and it's the wiring basically. And you can see a normal eye in the top, and we measure something called cup to disc ratio. And in a normal eye, you can see the cup is very small. If you remember back that first slide where we saw the orange eyeball on the right, and the image of that is a little tiny light coming in there, and then the glaucoma one had this big hole in it. That's the bottom picture, glaucoma. You can see that the number of nerve fibers have died. So the game plan here is you have to try to protect what remains, because retinal ganglia cells don't regenerate. They're nerve cells, and neither do retinal pigment cells. Next slide. So what our colleagues at University of Miami, Bascom Eye Institute have discovered is their particular receptor. It's called the EIR receptor, and its job in our body, and it's in a number of cells in addition to the retinal ganglia cells, its job is to control and manage the signaling down the nerve of the electrical signal back and forth. And when it's upregulated, meaning turned on, it directs the signal in one direction. And when you turn it off, it directs the signal in another direction. In the case of the eye and the retinal cells in the eye, what's going on here is when they become inflamed or under oxidative stress and they start to be damaged, the cells will die. But if you antagonize the EIR receptor, meaning turn it off, it begins to preserve those cells and protect them. And if you look at the four panels, our two leads, 1002, IR 1003, versus the control, So the native means that's what it normally looks like. And what you're looking at are the retinal ganglia cells, the nerve cells of the optic nerve there, the green fibers. And then you see when we damage the nerve, there's no protection. All the cells are dead. That's the vehicle control. Then when you look, if we treat first and we then damage, we can protect those cells because you can see a lot of them. It's roughly more than 50% of the cells remain, which is quite encouraging. Next slide, please.
About 7 minutes.
Yeah, we'll get through this. Okay, so I'm going to just go over this slide quickly. What we're showing here is not only do we keep the cells going, but the wiring. We help protect the wiring as well, the so-called axon and dendrites, little whiskers, if you will. Next slide, please.
Yeah, but there was a question about Avastin. It says, is Avastin used to treat wet? It's used to treat wet eye. Does it do anything for dry eye if the patient has a
No.
wet and dry?
No.
No. Okay.
Wet is called neovascular. So it's the ingrowth of blood vessels in the back of the eye. Dry is all about this lipofuscin. It's atropic. The cells are dying from atrophy due to this toxic substance that's killing them off. Next slide, please. And it should be good for, let's just keep on next. So data summary, next slide. So our pipeline, we're moving into the IND filing, and we should hope in the next 12 months to be commencing the phase 1, phase 1B study. Next slide, please. Tony, maybe you'd like to talk, and let's forget the clinical trial design. Next slide. Okay, Tony, why don't you take it over?
Yeah, no problem. It's very difficult to give you a full picture in 20, 25 minutes. So you have to kind of stick to things that are, recognizable and that you can understand. Martin and I have a background that's allowed us to take next generation technologies. We've worked very closely with the NIH, the National Institute of Health. When we started Iovance, was through the National Cancer Institute, which is part of that. We took a cell therapy, went from bulletin board to NASDAQ. It got up to a $7 billion market cap. How's that happen? Doesn't happen because Martin can talk and I can talk. He's actually more intelligent than me. It happens because we find assets that are actually prosecutable, that can actually go through the rigors of going into an FDA clinical trial, and that capital markets people like yourselves can understand and see where the value, there's a number of value points that go on from there. We went from there to, we started a company called GT Biopharma, which we took public from a bulletin board company to an uplisting on NASDAQ, February 16th, 2021. Within 90 days, we were a Russell 2000 company trading at $20 a share with many millions of shares. Again, how's that happen? It's data. You don't it's all about, in biotech, if you're not talking about data, you shouldn't be listening. All right. So we spent that. It's also the people that are involved with it. Our relationship with Cornell Weill University is, it's not just a traditional biotech. We license the technology. Of course, we have the licenses. They're actually owners in our company. They elected to take, they own 4% of our company. The same thing happened with the University of Miami Bascom Eye Institute, because they also, instead of going ahead and, wanting to just have a traditional relationship, they also own 4%. So that means we're on their portfolio list. That gives us a lot of credibility. And how do we get those kind of relationships? Because we've been there and we've done that. All right. You can only do it through hard work, no excuses, and execution.
Do they introduce you to their LPs also? So in other words, University of Miami Endowment or their healthcare system is invested in several different other VCs, PE firms, et cetera. Do they introduce you to their LPs?
Yes.
I mean, that's something that pension funds do all the time.
Yes, not only that, but they've made introductions to a large pharma. So yeah, they are, it's different. Having done many types of agreements with universities, if you're just a licensee, it's, you're just paying money really, right? It's when you cross that bridge and you enter into not only the license, but sponsored research agreements. So you do some of the work at the university, but you also get them to be equity partners. Now you become almost like you're in a venture portfolio. They do treat it as a venture portfolio. And then of course, then They make introductions for you. If the milestones slip, they're obviously much more aware and helpful. And certainly on the front end of the deal, giving them equity in the company lowers your royalties, your upfront costs, and lengthens, you get more time. They're not as hyper aggressive as typical.
That's good. They have aligned interests. Great.
Yeah, they're shareholders, just like the rest of us. And in our case, as Tony said, Miami owns, I think 4.25%, and Cornell owns 4.25%.
And the other thing we've done, we've actually filed our DRS with the SEC to go and do an IPO. We have a bank, all right, ready to do that with us. So the toolkit's being put together. We've been through the process many times. And that also speaks to value, right? Is when do you get involved with a biotech company? If you know that you can prosecute these assets, you can get it into the clinic, you start driving data. Martin's been very, very good at, even in our phase 1 clinical trials, we've started seeing signaling and things like that in the past companies that we've had. So our progress is geared towards building a public company, and a public company is really two businesses. It's a public company and you have to spend your time and your money doing all the things that you have to do for that. But in our case, it's also a biotech company. They have to integrate, and you've got to be able to take that story and where people can understand what the value creation is going to be. So we've done that a number of times, and we're very excited about what we're seeing. The data that we're seeing from Cornell and from Miami University is exciting, and it's different than what's out there right now. We're not in Cornell. What we've seen, and we think it's translatable, is that the ability to not just slow the disease down by 20%, but perhaps even arrest it. Now, the proof is always in the pudding, right? So any other questions? Yes.
So about 20 years ago, I was driving down the street and I see some black rain in front of my eyes. And turns out it's retinal detachment, 850, refractive error. So they sent me my seeing eye dog to Vietnam. But then my son gets the same thing. That's 30 plus years younger. So what's happened though, I think, is that there seems to be, at least for high blood pressure in the eye, a drop you take says you won't get macular degeneration wear out before then. So it seems like there's so many advances. Drugs, took out a cataract and I got a toric lens. This guy, Dr. David Chang, one of the best in the business. Now I don't have contact lenses anymore. So it seems like there's a lot of those. So given that there's all these problems, it seems like you should find a big audience on this.
Well, the market for AMD is they estimate it's going to be 280 million people that have age-related macular degeneration. 80% of that is dry. So that's $250 million market. It's huge. When people say, well, there are other companies doing it, I remind folks, like, there's Tylenol, there's Advil, there's Excedrin. There's plenty of opportunities out there for us.
250 million people.
Yeah, there's 280 million people. It's a huge market. And it's funny you mentioned that I had retinal surgery on my left eye as well. Do you see the little floaties? I had a slight tear in the eye. But the eye drops are for reducing the pressure, and that's in a glaucoma setting.
Yeah.
So not in the retinal tears, they take a laser and they stitch it, they tack it down. And so no problems in my left eye after that. But it's ophthalmology is kind of an interesting market. It's the big pharma are getting more and more interested with it. Heretofore, it was more of a specialty kind of deal. But now we see when you start talking about 280 million patients worldwide, and 80% of those 250 million patients, this for dry AMD, that's a big market for pharma, and at $1,000 a dose, you can all do the math. It's a huge number.
Glaucoma is 80 million people.
And you guys will be at dinner. We'd love to hear more about how the
Thank
deals
you.
you've done, this deal, and the deals you're looking at. So it sounds really great, and it's a great sector to be in. So thanks so much.
Well, thank you guys. We appreciate it.
Oh, thank you.